So for this talk today, we're going to talk about eye development. And some of the new ideas here are, this is the first time we really talked about an organ development per se. I guess unless you think of it as being an organ. But also, it's a good chance to think about what we have learned from Drosophila and how it applies to humans. So in Drosophila eyes, they're very different than human eyes. For one thing, they're not, well, they're composed of these 800 sort of simple eyes. So 800 relatively simple clusters of cells, each functioning as a visual unit, are stacked together in very regular looking arrays of cells, and together they make up what's called the compound eye. So it is able to sense light and these organisms are able to see, but the way this organ is built is very, very different than the human eye, which has its photoreceptor cells and layers of cells in the iris and so forth. So we'll do a little contrast and compare. And one thing to keep in mind is, do you or do you not expect to see the same sorts of genes involved in building an eye for Drosophila versus building an eye for vertebrates? We will answer that question. Okay. So the eye in vertebrates is going to come from an ectodermally derived tissue, which is why we're talking about it now. And specifically, the eye tissue in vertebrates comes from this vesicle that's set up in the early embryo. In particular, the diencephalon is the one that will give rise to eyes in the adult organism. So initially, we had described that in the neural tube, we see these partitions developing, and we get to the stage where we have 5 different partitions. The second one from the anterior is called the diencephalon. And as time goes by, we'll see some changes in that structure that give rise to the first indication of cells committed to eye cell fate. So this happens as an outpocketing, so a bulging out of a little pit of the section of the neural tube within the diencephalon. And it makes these 2 structures that are going to be called the optic vesicles. So you see they have 2 little out pockets there. And then within this vesicle, a portion of cells would be set aside to become different sorts of things, such as retinal cells or lens cells. So a portion of them are going to become lens cells down the line. So this is more of a longitudinal section taken through the embryo along the anterior and posterior axes. Let's think about what this might look like in cross-section now. So cross-section at the level of the diencephalon. Here you see these 2 regions where the neural tube is pushed outward. And these structures, showing just one side here and out-pocketing, that whole thing is called the optic vesicle. So this then is going to, a couple of things to point out. First is that it's still in contact with a layer of ectoderm on the outside, right? So the neural tube is internal structure to the embryo. It's covered up on the outside by a whole layer of ectoderm. And in the diencephalon, that little optic vesicle is going to be in contact with a portion of the ectoderm that's very near to it. And so this then, hopefully it's triggering some thoughts that, oh, maybe some induction is occurring here. Maybe some paracrine factors or maybe even some juxtacrine factors might be going to be important in this process for specifying what the ectoderm would become and what the adjacent optic vesicle would become. And both of these tissues do actually contribute to part of the adult eye structure. Then another thing to point out is the cells in this area change their cell shape, and they take on this kind of flattened, elongated cell shape, and this structure is called a placode. And that's more of a general term that you'll see in development anytime you get a localized thickening or flattened area of cells. So it's just something we can see morphologically, so we call it a placode. So we're going to have a lens placode, and then below that, a neural placode. So this optic vesicle will then then continue to reshape itself and take on the appearance of more of a cup-shaped structure. So this will be called the optic cup, and it's very easily recognized within the embryo. I'll show you a couple pictures from EM. And then after we have the optic cup forming, then this lens placode and the optic cup sort of fold in together, and that creates this structure here where we have a little bit of infolded lens placode, and then the optic cup really looking like a cup at this point with two different layers to it. And eventually we'll see these cells here from the ectoderm pinching off and becoming the lens of the organism, the lens of the eye. And then within the optic cup, two different layers in dark blue. The inner layer becomes the neural retina. So that's going to give rise to a lot of the cells within our actual retina, like the photoreceptor cells or bipolar neurons and so forth. And then we also have the outer layer that's going to contribute to pigmented retina. So just another cell type within this region of the eye. Okay. So, these structures that form early naturally go on to become the part and parcel of the more elaborate adult structure. So here, fast forwarding in time a little bit, now we've seen that lens vesicle actually pinch off. So in principle, it was kind of similar to what happened with the neural tube, right? You had an infolding, a pinching off, and a resealing over. So now we have a little ball of cells here called the lens vesicle. These cells will grow over time, and eventually they no longer have a hollow interior, but they're just a cluster of localized cells there. They start making a protein called crystallin, which is a protein that's very common inside of lens cells. And the crystallin protein can interact and form these arrays of protein. In fact, they eventually make so much crystallin that the nucleus of the cell sort of dies off or degenerates, and you get just these cells that are badged of crystallin, and they're all arranged there. And this gives the lens the properties that it does have of being able to refract light in particular directions. So this is a structure that is built out of proteins that originally came from cells. Then we also have some continued differentiation within the rest of the optic cup. So we're going to see some fluid develop in here that is becoming the vitreous body. So that's the internal gel-like fluid or substance inside of the eyeball. And then these cells here, the neural layer and pigmented layer of the retina, have some further differentiation to do to set up the final layers of cells within that. Then also we have a portion of this tissue. This came from the diencephalon, which was part of the developing brain. And so we have already a handy stock of cells that go directly back to the brain. So that will become the optic nerve, which, as you know, is going to be the place where the neurons are located that transmit this visual information back into the brain so that the brain can understand what we're seeing. Okay, some pictures. Here is embryos. I think these must be mice. And we're seeing a first development of these optic vesicles. And you can see how there is a layer of ectoderm on the outside of all of this. So optic vesicles in close proximity to the ectoderm. Then thickening of these cells here in the lens placode. You can see that, again, right next to the optic vesicle. Here's a little bit later stage where the optic cup really looks like a cup. And here's the lens vesicle just partitioning in. And then finally in D here, the lens vesicle has continued to differentiate, and this will become the lens. Then I'll also point out here we have still this outer layer of ectoderm, even after the lens vesicle is pinched off. This outer layer of ectoderm is going to contribute to the cornea, which is a thin layer of cells on the outside of the lens that covers it up. So, as kind of a reminder, I'm sure you're encountering this somewhere along the way in a physiology class. This is the adult eye in the mammal, and some of the structures are the cornea on the very outside, The iris is the colored part of the eye, and that actually can expand and contract in that center region where there is no iris. That gives you the pupil. So this is our colored part that might get larger or smaller depending on how much light is present at the time. Then behind that, a layer of cells that are the lens, and they're refracting the light into this vitreous body. And then the retina cells are in the back, and this yellow layer is going to contain some of the cells that are contributing to the retina, and then finally all of these will, these neurons contribute to the optic nerve. Okay, so a new concept to talk about at this point is the idea of reciprocal induction. And the more that we get into talking about organs and how they formed, we're going to see this concept come up right, left, and center over and over again. And it's pretty similar to what we've talked about so far with just regular induction, only this takes it sort of one step further. So in the case of reciprocal induction, we start out with an induction event, so one type of cell signaling another cell to try to evoke some sort of response or change in that neighbor cell. So first guy speaking on the telephone to a second guy. Then what's also needed to complete the thought here is that the responder has to be able to receive that signal. So they have to have the appropriate receptor, maybe they have to be in the appropriate state of specification. They need to be able to be confident to respond to a signal coming in, and not all cells are. Maybe only cells with the right place, the right time, the right state of differentiation could respond. So one guy can call, but the other guy has to pick up the phone and actually listen, and maybe do something about whatever message he receives on that phone. So then, to take this that step further, the reciprocal part is that now this layer of cells that received information and was changed by it, they become the inducer. So then they send out a signal now to the first guy and tell him, we want this change, get on the ball. So he has to be the confident one to respond to the signal. And you can see these multiple waves of cells, layers of cells, talking back and forth to each other and changing each other and sending out chemical cues and responding to those chemical cues. And so it's a good mechanism for cells to continue to be in an interaction as differentiation proceeds. And we'll see this used a lot. So as some examples applied to eye development, we can first see as the lens vesicles form and pooch out from the diencephalon, they're initially the inducer. So they send out some signal to the ectoderm that happens to be overlying that particular portion of the embryo, and tell it, why did you guys get together and form a lens placode? So they respond by changing their cell shape and provide that little localized area of cell thickening. And this happens with a certain typical timing within the embryo that you see optical vesicle forming, and then shortly after that, due to this induction event, a response on the part of the lens placode by day 9 in development. And then, at this point, now the lens placode is going to become the signaler, and it's going to specify the next step for the optic vesicle, maybe to start changing its shape. So how do we tease this apart experimentally? What can we do for show it, move it, or block it experiments to try to learn something about this system and the timing of it and how it's happening? Who is the inducer? Who is the responder? This is some ideas of experiments that could be done in, say, frogs or maybe even mice or maybe chicks. And you could start out by saying, okay, we have this observation that we can see lens vesicle next to some ectoderm, and we see that that ectoderm typically responds in a certain way. So if we want to test the interaction between those two, one thing you could do is go in and cut off a donor piece of optic cup or optic vesicle and place it under the ectoderm in a different part of the embryo. So testing whether this embryo or that ectoderm could respond to the signals there. How far does that confidence go? We also testing the idea that this is actually the source of those initial paraffin factors that might be an inductive signal. And so here they found out that, well, if you put this anywhere else, that ectoderm is not competent to respond, or at any rate, we don't see a lens placode forming in any other region. So then you can follow up on why that might be. So if you, say, remove the optic vesicle by just going and taking it out, removing the source of the signal, now what would happen to the lens? Nothing. The lens is not induced. So we need that optic cup. It's necessary for that next step along the line for lens placode induction. And you could also do mix and match types of experiments. You could take some tissues that are not optic vesicle, but maybe, I don't know, other parts of the diencephalon, other parts of the neural tube at that point, and see if they have any ability to continue to move this process along to get lens placode forming. And in general, no induction has occurred for those events. So it really looks like we have a very specific area and response and confidence and back and forth with these particular tissues. So after we induce the lens placode, now here we have lens placode becoming the inducer, so a reciprocal induction event. And that stimulates the early optic cup to infold in this way and start to become more cup-shaped, differentiating an inner layer and an outer layer. And then once we have the lens capsule or lens vesicle forming, those become a source of inductive signals for both the overlying ectoderm to become cornea and for this first inner layer to become more of a neural retina. So multiple waves of this as development proceeds. So within the retina, we have neuroblasts. So we have these sort of committed precursor cells, if you will, cells that are still in a fairly stem cell-like fate, but at least they know they're going to become neurons or glia. And ultimately, this will become organized into a very tidy, layered fashion with different groups of cells. So from the pupil going into light, you would think that it encounters photoreceptor cells first, but actually the photoreceptor cells are more at the back of the eyeball. And they have this array here where they have rhodopsin or light-sensing pigments within those cells. And then they can sense whether they've encountered light, and they communicate that to another layer of cells, then it's the bipolar nerve layer. And then those form synapses on the ganglion cell layer who send their axons back into the optic nerve. So we have at least 3 groups of cells here that are involved in light transmission, and they're all organized in terms of where are their processes versus where are their nuclei. And then if you stain through these sections, you can see how regularly organized and tidy it all is. So this is something also that is set up during development and contributes to the functionality of the organism. And this happens over time. So initially, within that layer of cells in the optic cup, you might see a lot of cell proliferation, but things are not really organized into layers yet, or even into neurons. But these begin gradually to separate out and set up different layers with the different cell types differentiating into bipolar cells or photoreceptor cells or so forth. And so, you would expect this whole process, even this segregating out and this differentiation into different phenotypes, all of that would be controlled by genes. So you could probably find mutants where the process would get so far and then things would kind of break down and you don't get enough cells of particular cell fates. Kind of a new finding in the field has to do with the very close spatial and temporal regulation of genes expressed in this process. And, I think we often make the assumption that if a gene is transcribed into mRNA, that we're definitely going to get some protein made out of that as a consequence. And not always. Oftentimes the mRNAs are regulated as to whether or not they can be translated into proteins. And here's an example of this in eye development. And this happens to be the frog. So within this layer here, we see a couple of genes. X stands for xenopus, which is the frog, and then the name of the gene, so this OTX5B. The mRNA is expressed pretty broadly, but if you look where it's being translated into protein, so in situ hybridization versus immunohistochemistry, the protein only actually is made in one select group of cells at this time, and that is associated with the photoreceptor cell fate. So this is early-ish on stage 34. Then, fast forward to stage 37, so maybe only a few hours later in development, that same mRNA is fairly broadly expressed, but a different group of cells is translating it into protein. And at that point, it's more involved in the bipolar interneuron cell fate. So another group of cells, close proximity, different job in life, and it's all regulated by the timing of translation of this mRNA. So, that's kind of exquisite level of control here to, make the mRNA and then just decide exactly when and where you're going to translate it. So when it comes to translating mRNAs, we're coming to appreciate the function of another big group of genes that up until a few years ago, we really knew almost nothing about, and these are the microRNAs. And it's not just microRNAs, but there's other types of genes within the genome that are scattered around, and many of these only are ever translated into RNA and never into proteins. The RNAs that they translate to have functions in regulating other gene products in the cell. For example, one thing they do is they may go and bind to specific target mRNAs and regulate whether that thing survives and or whether it's translated into a protein. And here's an example of that. This, I think, comes from the worm C. elegans. So there is a Lin14 mRNA that's encoded within the genome, and it has several different exons and introns. And then there's also a Lin4, so a totally separate gene, and that thing makes a microRNA. So then in the cell we have these long Lin14 mRNAs plus the Lin4 microRNAs, and they bind together. And especially at the untranslated region of the long mRNA, you'll see these Lin4s pairing. So here's several examples of where the Lin4 would actually pair with the Lin14 mRNA. And it's kind of interesting because it's not a perfect pairing, which, apparently there's enough sequence recognition that you could actually get some specificity to this. But there's some little bubbles out here indicating that these are places where the nucleotides are not complementary and therefore they're not going to pair. However, when you see the Lin4 pairing to this, it tends to inhibit the translation. So there may be a time during development where Lin4 is made and it is present and inhibiting, or a time in development a few hours later where those Lin4 microRNAs have now degraded and the inhibition is released, and now we translate the data. So kind of keep your eye open as you go on in biology to hear more about these microRNAs or other RNA type of regulation. Okay, here's just a picture of this for a vertebrate where we have a microRNA that is acting at different stages. So initially there's a lot of this microRNA present and we're seeing relatively little translation, but then at later stages it is gone and you get more translation. So timing becomes really critical for all this. And even just an hour or 2 hours could make a big difference in exactly how that cell fate is unfolding for the embryo. Okay, I want to pivot now and talk about Drosophila. And I actually did Drosophila for my PhD work, and I really liked working on this system. One of the great things about it is you have this tremendous flexibility in terms of the genetics. So you can do mutant screens, you can get back a huge variety of different types of phenotypes, and find even really subtle mutations that give you a big group of genes, and you can just learn a ton about processes like eye development from the Drosophila system. And this is just a snapshot of some of the different phenotypes you could see in Drosophila eyes. For one, you can affect the color. So there's, I'm probably guessing 20 or so different mutants with slightly different varying shades of color. Some more obvious than others, as you probably saw during your Drosophila lab. We can also affect overall size and shape of the eye. So this may be teaching us about genes that are needed for organization or proliferation of these eye cells, or how they interact together to establish the even surface of the eye. And then this one, this is what's called the rough eye phenotype. And what you see for the wild type up there is these 800 ommatidia with a little bristle in between each one. They're very, very regular and beautiful and almost like a lattice. That's how, organized they are. But that makes it that if there's any kind of disruption to this, even if it's really subtle, you can still see it. Because, you can obviously tell that this is not the even pattern that you can see in the wild type embryo. So there's this ability to pick up even really subtle mutant phenotypes, and therefore to really understand how genes are interacting and impacting each other in this whole process. So one interesting mutation actually just eliminated the eye. So the rest of the fly is totally fine, but there are no eyes apparent. And even if you do electron microscopy, you scan across the lovely fly head and you see no evidence of ommatidia or eye development at all. So for this, since we need to name this gene, whatever it is that is mutated here, we need to name it after the mutant phenotype. This gene, or this mutation, was called ILIS, or the ILIS gene phenotype. And when we eventually discovered what gene this was and sequenced it, the gene was given the name of PAX6. And the PAX stands for a conserved domain that's present within this family called the paired box. So PA for paired and X for box. So PAX. And there are a number of different genes that have a conserved region. So this is a distinct and unique conserved region found among all the PAX gene family members. I've shown it in red here. This PAX6 also happens to have a homeobox. So they have a distinct and separate region of the coding region that is very, very similar to all the genes that have this homeobox family. So it actually has two, and the sequence of the paired box and the sequence of the homeobox are different, right? Okay. So what is this thing? It folds up, and it is a transcription factor. And the homeobox region may be the portion where it interacts with some targets on DNA, and the paired box region, that could be involved also in DNA recognition and or binding to other protein partners. So these are the two things you can keep in mind for transcription factors. What other proteins does it bind to? Mainly other transcription factors. And what are the DNA targets that it's going to find? So what specific sequences within DNA enhancers would it recognize? So the fly has a PAX6 gene, and the mouse has a PAX6 gene, and humans have a PAX6 gene. And these are the phenotypes that we see. So for fly, depending on if we completely obliterate PAX6 function or only partly, we can have eyeless or very small eyes. In the mouse, a heterozygous situation, so mutant allele plus wild type allele, heterozygous, that gives you a small eye phenotype for the mouse. Or if you have a doubly homozygous mutant, it's no eyes altogether, and it also is lethal. Now, it may not be lethal due to the lack of eyes per se. It's probably due to PAX6 being involved in other important tissues that you would need to survive. In humans, PAX6, so we combine situations where there's a heterozygous situation, and this acts as a dominant mode of inheritance, so even heterozygotes have a phenotype. And what this is aniridia, which is affecting the development of the iris. So it's not like the whole eye is gone, but in the process of building that eye, clearly something wasn't patterned quite correctly. So partial or complete abscess of the iris. There was one incident known where there was a fetus that was homozygous for PAX6, and that was lethal. Again, probably because PAX6 is needed for other functions in the embryo as well as eyes. But this is interesting. Think about this paired domain and the homeodomain. If we did a blast alignment and we look at how the amino acids match up between fly and mouse or fly and human, they're unbelievably well conserved. So fly and mouse, we're talking insect and mammal. Within this paired domain, 94% identical, which is like outrageously identical. It's almost exactly the same. Same thing in the homeobox, 90% identical. And now human and mouse, two mammals together, well, they're actually 100% identical. And just within these really special domains, not the whole protein, but within these regions. So these are the critical regions. So in Drosophila, if we mutate PAX6, we get eyeless. What about in mouse? So here's a PAX6 knockout mouse. So this is like a golden opportunity to have a gene of interest and go in and do reverse genetics and make a knockout mouse and see what happens. And so here is the slice through the developing brain, forebrain and midbrain, and right at the diencephalon we should be seeing some nice optic cups. And we see beautiful ones here in the wild type. And over here in the mutant, there are perhaps at least some initial optic cup forming, but it doesn't progress into the fully differentiated phenotype with the 7 layers of organized cells like we see in the wild type. It's just this kind of stunted lack of eyes. So here's a newborn mouse, and it's a bit hard to see, but there's a little small slit here. That's the functional, fully formed eye. It's just not quite open in this newborn mouse. But in the PAX6 knockout mouse,